Okay, so thank you everyone who was brave enough to come here despite the rain. And thank you to everyone who is joining us online. I guess you might be able to hear us. We definitely cannot hear you. And today it's my pleasure to introduce Tim Petros from NICHD. He is our NLM Colloquium speaker, and many of us do know Tim as an active contributor to the NLM DR program. He helped us a lot with his work and insights during this summer and fall. And going to the background, Tim earned his PhD from Columbia in 2009. After that, he was a postdoctoral fellow at Cornell and NYU. And in 2007, is everything okay? In 2007, NIH, in particular NICHD, has been very fortunate to bring him on board as a tenure-track investigator. And Tim's work, which has started, I believe, initially with mass spec and interneuron differentiation, has expanded into the field of genetic and epigenetic mechanisms that regulate early interneuron fate decisions during neurogenesis. And the results of Tim's group's work has been published in a lot of leading journals, including Neurons, Nature Genetics, PNAS, Development, and several others. In addition to that, Tim has received multiple awards, including the Division of Intramural Research of NICHD Scientific Directors Award, Mentor Awards, etc., etc. The list is way too long to mention, just enough. Today, the title of Tim's talk is Mechanism Regulating Fate and Maturation of Forebrain Inhibitory Interneurons. And before we jump into that, I would like to briefly talk to those of us who are attending virtually. If you have, I hope you can hear me, or you will be able to see the presentation. If you have any questions, please do send them to the email address that hopefully will be provided to you by Quinn in this video chat function. I'm not sure how it works, but if you see the email address, please feel free to send your questions there. If not, you can also use the feedback button on the videocast page. Those questions will be delivered to me somehow, and I'll ask Tim these questions at the end of his presentation. Also, if you are interested in joining us for lunch at noon, you are more than welcome. We will go to the cafeteria at Nature, which is fortunately open today. With that, it is my pleasure to welcome Tim to our NLM Colloquia.
Okay. Thank you for that kind introduction. It's nice to see some familiar faces here. I really enjoyed my time on the tenure track search committee. I learned a lot about NLM and what you guys do as an organization, and again, I'm very happy to present my work here. So, as the intro said, my lab focuses on mechanisms that regulate fate and maturation of GABAergic inhibitory interneurons, and this is one of my many failed cover image submissions, but basically on the left is a section through the embryonic mouse brain, on the right is a section through the adult mouse brain, and so my lab tries to understand how you go from this small population of cells in the embryo to generate this incredibly diverse population of GABAergic cells that populate a variety of brain regions and are critical in nearly every aspect of brain function. So it goes without saying that brain development, in particular human brain development, is an incredibly complex and prolonged process, starting with the neural tube structure, and then over months and years forming this complex structure that we all try to understand, and how you generate this complexity and diversity, I think, is one of the most exciting and challenging questions in biology. Now, if you look at comparison of species of different brains, you can say up here is the little mouse brain that we use to model system, and over time, you eventually, evolution has given rise to the human brain with over 86 billion neurons that makes trillions and trillions of synapses. And then you can also look at cross sections here. And again, here's the mouse brain. So even though we consider it a model system and more simplistic, it is still incredibly complex in terms of its cellular diversity and connectivity. But there's still a lot we can learn about it and hopefully apply to human brain development. Now, as I said, the cells, I study these inhibitory interneurons. The brain contains excitatory cells and inhibitory cells, and this balance is critical for maintaining normal brain function and circuit function. And so these inhibitory cells are born in the ventral part of the brain, the bottom part of the brain, from these ganglionic eminences. And here it is kind of in a 3D view. There's the lateral, the medial, and the caudal ganglionic eminence. And most of my work is focused on the medial and the caudal ganglionic eminence here. These cells are born, they become post-mitotic in these regions, and then they go and migrate and populate all regions of the forebrain. We know a little bit about some of the genetic mechanisms, the genes that drive interneurons from these two regions, the CG and MG. Cells that arise from these regions form different types of interneurons, and they're more or less non-overlapping. So depending on what mature type of interneuron you are, we know whether you arose from the MG or CG. So these two regions give completely distinct types of interneurons. And again, as I mentioned, they populate the entire brain in different densities and different morphologies, and are involved in pretty much every aspect of brain circuitry.
What is this flavor, please?
In the previous slide, let me go back. So this is actually in utero electroporation. So this is me labeling cells here with just a GFP plasmid that then expresses GFP, and then 30 days, about 40 days after the electroporation, this is where the cells have migrated and populate. So it's just a reporter of cells that were born in this brain region. So if we zoom in on these cells, here's actual reconstructions of different types of interneurons, and it can give you a feel for the striking different morphologies. Some basket cells have very dense axonal arbors and synapse very locally, and there are other cell types like this Martinati cell, which sends its axons further away from the cell body and forms synapses on distal dendrites. If you look at this cartoon, you have this very boring pyramidal excitatory cell here in gray, and all the colored cells are the different types of interneurons that can synapse on these excitatory cells. And so this gives you a flavor for the different morphologies, connectivity patterns, and again, how do you generate this incredible diversity of inhibitory cells from basically two brain regions in the embryonic mouse brain? Now, what's interesting is that interneurons make up only about 20 to 30% of the neurons in the brain, and the excitatory cells make up 70 to 80%. But if you actually look at the different subtypes, so this is from the Allen Brain Institute, where they've classified subtypes based solely on transcriptomes, you see in the mouse cortex, basically 50% of the different cell types in the brain, defined by gene expression, are interneurons, even though they make up a smaller proportion of the cells. And this actually expands in the human cortex, where about 60% of the cells of genetically defined cell types are interneurons. So there's incredibly higher diversity compared to the majority population, the excitatory cells. Now, dysfunction of these cells is associated, or in some cases causative, with a wide variety of neurodevelopmental diseases, most notably something like epilepsy. If there's too little inhibition in the brain, that can cause the brain to be hyperexcitable, and that can lead to epilepsy and seizure disorders. But interneuron dysfunction has been noted in a wide variety of diseases. And in fact, many genes associated with these diseases are enriched in neural progenitors, so immature cells, and oftentimes actually in immature interneurons. And then what my lab has become more and more interested in is the growing evidence that many perturbations and mutations actually affect epigenetic machinery, so gene regulatory mechanisms rather than the gene coding sequence themselves. And so this is kind of a a driving theme of our lab, that we believe that we need a better understanding of both the genetic and the gene regulatory mechanisms that guide these initial fate decisions early on in the embryo, and that should increase our understanding of both normal development and various disease etiologies. So when there are perturbations in these genes, what goes wrong early on that eventually manifests into a disease phenotype. So I'm going to tell you about a couple stories today. The first is we want to basically define what we call an Epigenome Atlas that basically set the ground truth for how these cells look from these different brain regions in the embryonic brain. And then once we have this baseline, we're able to see how perturbations affect this development. So I'm going to tell you about a variety of techniques that we use to basically kind of define the ground truth of these cells, many single cell sequencing assays, as well as some bulk sequencing assays. And then once we've established this, this now allows us to perturb the system with genetic mutations to understand how alterations of either gene repression or gene activation can affect cell fate, cell function, and ultimately animal behavior. And then lastly, if I have time, I'll tell you about some new data looking at one specific transcription factor that's highly correlated with neurodevelopmental disease and what some of the insights we have from our most recent studies. So a lot of our experimental workflow follows this paradigm. We have our embryonic mouse at a particular age we're interested in. We can then open up the brain, hemisect it, and look inside. This is obviously stained and colored, but you can actually see these structures just by eye without the staining. They're physical bumps into the ventricular space. So it's very easy to access these and dissect out the MG or the CGE. And here it is in cartoon form. So we can dissect out these regions, prepare single cell or single nuclei suspensions, often FACS sort them. We can put them through our 10X system to do single cell sequencing, or alternatively do some bulk sequencing with some other assays, and then use the NICHD sequencing core. And then what my lab spends most of their time doing is the bioinformatics, figuring out how to massage this data and manipulate it to try to understand biology from these very complex data sets. And this is a fairly accurate representation of what my postdocs think about day to day as they work on this data set. So there's been a lot of work that had looked at transcriptomes, so gene expression profiles from these embryonic brain regions. But we wanted to kind of look at one step higher to look at chromatin accessibility and start to understand how the chromatin looks in these distinct brain regions that give rise to distinct cell types. So a couple years after I joined here, 10X had developed the first commercially available single-cell ATAC-seq platform. Previously, you could do bulk ATAC-seq to look at chromatin accessibility, but in heterogeneous environments such as the developing brain, it's really not that informative. So what ATAC-seq is the assay for transposase-accessible chromatin. Basically, you use a transposase enzyme that can go and identify open regions of chromatin. The transposase can find these. It has tags. It can basically cut this DNA so that you're left with these little pieces of DNA that represent open regions of chromatin, and then you can sequence it with these tags. And so we performed single-cell ATAC sequencing on these 4 brain regions. So we dissected out the cortex, the LG, the MG, and the CG. And again, these regions give rise to distinct, non-overlapping types of interneurons. We prep the samples, and then basically you have the readout where you have sequences, sequence reads throughout the genome, and what you're looking for is regions where there's a high amount of sequence reads in a variety of cells, and then that would basically be above a threshold that you would call a peak, and this is a region that is accessible in the single cell or the group of cells that we're looking at. And so what you see here is a UMAP plot of this data. It looks just like a single-cell RNA-seq, except in this case, each dot you're looking at represents chromatin accessibility rather than gene expression. And you can see that the cells from each region kind of cluster together. You have excitatory cells in the cortex, MG cells over here, and then LG and CG, for which we know less. There's less distinct differences in gene expression profiles. They actually kind of cluster together here. And you can perform different types of analysis, just like you could with single-cell RNA-seq. You can have putative cell clusters. In this case, we know kind of about how these cell divisions work, and we can kind of define different progenitors in here. And then you can also do pseudo-time to look at the development of these regions and these cells across neurogenesis. Now, in this case, what we're looking at here is heat maps showing regions of promoters of various genes that are accessible. So nestin is a gene that's expressed in radioglial cells, very immature cells, and you can see that the accessibility of the nestin promoter is restricted to these cells that are these radioglial cells, and as the cells go further along through development, this loci gets shut off and becomes closed. And so you can do this with various genes that change over time, and we can look at it at various genes that have spatial expression. So Nkx2.1, that I'll come back to a couple times, this is an MGE-specific gene. You can see that the promoter is only accessible in the MGE, and it's basically closed off in the other brain regions. And then there are other ways to look at this. If you look at the putative clusters, of which there was about 25, you can look at chromatin accessibility in clusters here based on their spatial location. So you can see, again, the Nkx2.1 promoter is mainly accessible in the MGE and absent in these other clusters. And again, you can look at that both in spatial divisions and along developmental time axes here. Now, this is kind of a fairly complex image, but it's a pretty good summary of how the differentially accessible peaks look in our data set. So in this case, the tissue is broken down by these color bars up here. These are different stages of neurogenesis here. And what we did is basically took the 25 or so clusters we had, putative cell clusters, and did unbiased hierarchical clustering so that we could identify the clusters that were most closely associated with each other. And so what you can see here, if you look in the upper left, there's 4 clusters here. These are all cortical apical progenitors, so they're all fairly similar. And you can see that there's actually some similarity between the differentially accessible peaks in these different clusters, and that makes sense because they're all very similar cell types. Conversely, if you look at differences between different brain regions and different cell types, you see that there's a lot less similarity here. So what we see is that the accessibility profiles of these cells actually matches pretty well with both gene expression, and there's quite distinct differences both between brain regions and between different cell types in development. So to take one brief tangent, as we were doing the single-cell ATAC data, we also in the lab generated single-cell RNA-seq data of the 4 brain regions, and we did this because we wanted to have both transcriptional data and chromatin accessibility in these brain regions. And so I won't go through all this, but we found distinct expression profiles in progenitor cells both between these brain regions, and here's some examples for LG, MG, and CG, and then also some really exciting entrances where within a single brain region, such as the CGE, you see one gene that's enriched in the dorsal CGE and one gene that's enriched in the ventral CGE. And so we hope and we think that these expression profiles in these different domains are critical for giving rise to different types of interneurons. But so we had the single-cell RNA data. Then we had generated our single-cell ATAC data. This was before multiome was available, which I'll touch on later. So what we did is we integrated our single-cell RNA data and our single-cell ATAC data into the same computational space. So while these are different cells, the RNA and the ATAC, they come from the same regions, the same time points. And so this allowed us to kind of look at how RNA and chromatin accessibility changes over time and identify differences between brain regions. And so just to give a couple examples here, what we saw is that in instances where genes were expressed earlier in the neurogenic timeline and then went down over time, we saw, so blue here is the RNA, red is the GAS, so that just is a fancy way of saying promoter accessibility. You can see that if you follow the blue here, the RNA turns off prior to the promoter and the enhancers shutting down. So it's as if the RNA is downregulated before the actual promoters and enhancers turn down. Conversely, if you look at genes that are expressed later in development, you see green and red, the promoter and the enhancers become accessible prior to the RNA being expressed. And that's what you would expect. You need to have promoters and enhancers open before you get RNA expression. So this is really nice. Now you can do it in multiome where you have the same data in the same cell. But this one example of the types of patterns that you can see when you look at these two data sets integrated together. Now this is great. Looking at accessibility and RNA expression is really informative, but it's only kind of correlative. So we can make predictions or assumptions about what's an enhancer and what's a promoter, but we really want to have greater data so that we can say with higher confidence, this is an enhancer of this gene and it's directly regulating its expression. So one way to do that is to look at histone modifications. So histones are proteins. The DNA wraps around these histones, and the histones can be modified in a variety of ways, phosphorylated or acetylated. And depending on the pattern of these modifications, that can be indicative of the state of the DNA. And so if we look at three of these marks in particular, H3K4 trimethylation is a good indicator for an active promoter. H3K27 acetyl is a good marker for an active enhancer. And then H3K27 trimethyl is a good indicator of a repressive state. And so we can take antibodies to these three modifications and perform an assay that's called cut and tag. It's very similar to ChIP-seq, but it uses more cells, or you need less cells to get good data, and it's much cheaper to look at, in bulk, these methylation patterns in these different brain regions. And so here's kind of a summary of how this data can look. So what we have up here is some analysis called Cicero, where we can look for individual cells. If a loci is open here, and then you go down, downstream or upstream, and there's another region here that is open in the same cells, you kind of get these peaks. And so this type of analysis is of interest, because if you're looking at a promoter and you see a region upstream or downstream that is often open or co-accessible in the same cell, that could be indicative of an enhancer. And so I focus on one gene here called ASCL1. This is a gene expressed in all the ganglionic eminences, but it's not expressed in cortical cells. And previous work had assayed this region right here as a candidate enhancer for ASCL1. Now, what we see in our data is that if you look in these four brain regions, this loci is certainly accessible in all the brain regions, but if you look at this marker for an active enhancer, it's actually strongest in the cortex and much less so in the ganglionic eminences. And again, in the cortex is where this gene is not expressed. And so when they looked at this enhancer, it actually did not do a good job of replicating gene expression, and we would argue that that's probably because of data like this, where the highest signal for an active enhancer is actually in cell types where the gene is not expressed. Conversely, if you look down here in gray, we found two other regions where you have really high signal for active enhancers and much lower in the cortex. And so we'd argue that by having this multimodal data set, you can do a better prediction for candidate enhancers for various genes. And so we have a whole list of candidate enhancers that we think could play important insight into gene regulation for various genes during development. So that's highlighted there. So another assay we did, so far we have accessibility, RNA expression, and histone modifications, but in order to have promoter-enhancer interactions, the DNA needs to come into contact. And so to really look at that, we used Hi-C, or more recently Micro-C, in collaboration with Pedro Roca's lab at NICHD. And so what Hi-C is an assay to look at direct DNA contacts between two DNA regions. And so if you just, so this is kind of a schematic of how DNA can look. Basically, to get an enhancer and promoter to come together, the DNA actually has quite organized structure, and there are various ways to bring them together. A lot of times they involve these CTCF sites that I'll come back to towards the end of my talk, to kind of segregate the DNA into topographically associated domains or tabs, and that's a way to restrict DNA from other regions and allow these promoter enhancer interactions to occur in specific ways. And so by using this Hi-C assay, you can identify these loci where you have distal genomic regions coming into contact, and again provides greater insight for how genes are regulated. And so this is how this Hi-C data looks. What you're looking at are these hotspots of higher intensity color in various loci. And then if you take a region like this, you follow it kind of down the side of the pyramid to here, and then come down this side of the pyramid. What that tells you, what this signal tells you is that you have a direct genomic interaction between this region here in the DNA and this region down here. And so if this happens to be at a promoter of a gene and this is a candidate enhancer, this provides further evidence that you have this genetic integration that you need. And so what we did is we performed these high-C assays on these 4 different brain regions, and then we looked at genes of interest to find regions where we saw differential DNA interaction. And so here's this Nkx2.1 gene that, again, is only expressed in the MG and critical for all cells derived from the MG. And what hopefully you can appreciate is at the MGE, you have this really kind of dark increased intensity here, and that's absent from these other brain regions. So this is a MGE-specific genomic interaction. Now, Capture-C is kind of a modification of high-C, but what it does is it confirms a direct interaction between, in this case, the Nkx2.1 promoter and a region downstream. And so what you see is this increased signal here, which indicates that this region has a direct contact with the Nkx2.1 promoter specific to the MG. You do not see it in the LG-CG cortex or liver as a control. And then you can integrate the other data we have, the single-cell RNA-seq, the ATAC, and the different methylations, and really, truly understand at a genomic level, a chromatin level, both how the chromatin looks, its 3D structure, where its interactions are, the histone modifications to tell you whether something is likely an active enhancer, promoter, or repressed, and then the accessibility in RNA. And what I'll note here is that this MG-specific interaction occurs with this gene called MBIP, whose function is not known in neurodevelopment. It's studied a little bit in cancer. But I did find a paper from about 15 years ago where they did staining for MBIP, and MBIP is also only expressed in the MG. It's not found in these other brain regions. So now we know there's a direct interaction between these 2 genes that are only expressed in the MG and not found in other brain regions. And then we went ahead, and you can do this for other genes and other brain regions, and there's lots of kind of exciting insight we have into region-specific, brain region-specific chromatin interactions in the embryonic brain, and to our knowledge, this hasn't been explored in detail yet. So we really think, we understand kind of the ground truth of how these cells look in the embryo as they're making these critical fate decisions. Now, I'll just kind of highlight what one ongoing study that came out of this specific Nkx2.1 interaction. As I mentioned, it's a master regulator of cell fate. And so we wanted to know, well, how critical is this interaction for expression and function of these genes? Is there a way to perturb this interaction to disrupt it and then see how that affects gene expression and cell fate? And why this is also relevant is because in human disease, there are variants of Nkx2.1 gene, and it gives rise to complex syndrome that can be called benign hereditary chorea, or actually the aptly named brain-lung-thyroid syndrome, because Nkx2.1, in addition to being expressed in the brain, is also expressed in the lung and the thyroid. And so patients that have mutations, variants in Nkx2.1, can express phenotypes that show dysfunction in these 3 organs with various severities depending on the patient. Now, what's also really interesting is that a handful of patients that present with this phenotype do not actually have mutations in Nkx2.1. Rather, they have deletions further upstream that encompass the MBIP gene. And again, there are no known functions for this in neurodevelopment. So now that we understand this interaction here, we wondered if it's possible that, that some of this phenotype that arises in these patients where the Nkx2.1 loci is intact, but the MBIP is disturbed or mutated, could be due to this critical interaction here. So again, how do we study this? How do we break up this interaction? Yes.
How do you distinguish between a gene-to-gene interaction versus an enhancer that might be related more Nkx2.1 and Mbip?
So that's a very good question and kind of complicated. So at this resolution, when we did this data at high C, we did not have the resolution to know whether the interaction, so all I can say here is this interaction is somewhere in this MBIP locus. You can see that this is a gene with 5 or 6 exons, and the resolution we had is we know there's an interaction somewhere there, whether it's a promoter-promoter interaction or whether there's a particular loci within an MBIP intron, that's an enhancer for Nkx2.1, or vice versa. We can't say anything about that at this level of resolution. The micro-C that I'll talk about a little bit does give you better resolution, where you can look at tens of nucleotides. And so then we can say with more confidence, okay, there's an interaction between the Nkx2.1 promoter and the MBIP promoter, or some intron in MBIP. But there's certainly evidence for promoter-promoter interactions that help expression of both genes. And then there's also evidence for enhancers in an intron or near another gene affecting the gene expression of another one. So that's really a question of resolution. How refined can you get to look at this interaction?
So, this is like a one-to-one interaction?
Yeah, so that's, again, it's about resolution. All we can say with this is that there's a direct interaction between some region here in this MBIP gene and the Nkx2.1 promoter. It's possible that there's 2 or 3 distinct interaction domains, but we don't have the resolution with this assay to directly answer them.
This interaction we have with the other chromosome also, like what you want, like an intra or interchromosome.
So you can look at interchromosome interactions with high C. So basically, what I've done here is focus on the specific region, but you can expand this out to all chromosome interactions. So if there are interactions between chromosomes that occur at a high enough frequency that it would kind of generate these hot spots, you could theoretically detect it in this type of data. But in this case, I just focused on this loci of interest where we have this gene and then a strong brain region-specific signal just upstream. Okay? Okay, so how can we disrupt this interaction? So this is where our collaboration with Pedro Roca was really critical. So in a study that we collaborated with, he was looking at the SOX2 locus, which is a really critical gene and interaction for the epiblast. And so basically you have a critical enhancer of SOX2, which is called SCR over here, and then the SOX2 gene is over here. And you have these little arrows are called CTCF sites, and as I mentioned, that's critical for parsing this genome structure so that you have this TAD domain here, so that basically genomic loci in here can only interact with each other and really are inhibited from interacting out here. So Pedro's idea is, well, what if we put in some artificial CTCF sites here to basically disrupt this interaction? And that's in fact what he did in this paper. You can do different kind of types or different organizations of CTCF factors. And I hope you can appreciate that when you do these modifications, what you do is disrupt, or in this case, minimize this interaction between the SOX2 locus and the enhancer over here. And then you can look at how this affects gene expression and phenotype. So we said, great, can we do this at the loci we care about on the Nkx2.1 promoter? And luckily it had these CTCF sites set up in a way that allowed us to do this. So Pedro designed some exogenous CTCF sites to insert in between these two genes with the goal of blocking this interaction. So this is new data with micro-C. It's basically a higher resolution, high C. And again, what you have is the Nkx2.1 gene over here, MBIP gene here, and in the wild type, you see this interaction. Now, in the mice, where we've inserted the CTCF sites, you now see that this interaction is gone. Instead, you have an interaction here, where you now have this blue CTCF site interacting with these red CTCF sites. So this basically was successful in perturbing this interaction between these two loci. Again, capture micro-C, another way of looking at it. Here you see this interaction normally over here in the wild type. It's now reduced in this mutant with an artificial interaction here. And so we've confirmed that basically we've blocked this Nkx2.1 MBIP interaction in these mice. So this is 3. So we had 3 going this way and 3 going this way. What Pedro's data showed is kind of the more CTF sites you put in, the stronger, the more robust phenotype you can have in terms of blocking that. And so these are human sites, so they don't interact, they can interact with these mice, but it makes it a lot easier for sequencing and downstream stuff. Okay, so what happens to gene expression? So this is in situ, looking at Nkx2.1 and MBIP, and what you can see in the mutant is that the MBIP expression is almost totally gone in the in the CTCF insertion. It looks like there's also downregulation of Nkx2.1. We're performing additional in situ to confirm this, but if you look at qPCR, you see pretty strong reduction, 50% or so reduction of MBIP in this mutant. It looks even stronger by in situs, and we see a slight but probably significant decrease in Nkx2.1. And we think this is really exciting because, again, going back to those patients, this could indicate that in these patients where you have mutations near this MBIP locus, what really could be happening is that you're disrupting this particular interaction that's decreasing gene expression and probably perturbing Nkx2.1 expression, even though the Nkx2.1 locus is intact, and thus giving rise to the phenotype. So we're still kind of working on this mice to look at changes in cell fate and any type of behavioral deficits, but this is a nice way of showing that here's a brain region-specific interaction we identified, and when you manipulate that, just this interaction, not the genes themselves, you can see changes in gene expression and ideally, hopefully, cell fate. Yeah.
I think we are supposed to be nice to people online.
Oh.
So it looks really, really strange to me. What you are doing, you are disrupting supposedly a regulatory element located inside MBIP. You are inserting a block in between that element and Nkx2.1.
Yep.
And you do see a decreased level of expression of Nkx2.1. Great. But you are not removing that element away from the host MBIP gene. And yet, the level of expression of MBIP drops down significantly. Are you saying that Nkx2.1 in turn upregulates MBIP, or how do you explain that?
So this goes back to your previous question. So is this an enhancer-promoter interaction, or is this a promoter-promoter interaction that they're both helping each other? So I can't say directly yes or no what the answer is. Based on this, I kind of think that these are two promoters coming together that are helping each other upregulate their expression, and now we force separation of this, and for whatever reason, MBIP is strongly reduced and Nkx2.1 is much less reduced. What I'll say is Nkx2.1, as I mentioned, is a critical regulator of all cells from this MG. So my best guess is that there's more evolutionary conservation there that is critical for keeping Nkx2.1 on. So I didn't mention it, I'll hop back real quick, but in that SOX2 locus study, what you see here is that despite the best efforts, there's still actually an interaction here despite putting in these sites. And what this paper showed is that it seems like evolution has found a way around various perturbations like this because SOX2 is so critical for development that any little mutation here or there, there's multiple mechanisms to help these interactions come together so that SOX2 expression is focused. And I'd argue that maybe the same thing is happening at Nkx2.1 because it's so critical for brain development, for lung development, for thyroid development, that I think there are genes that have this evolutionary conserved mechanism so that even if we try to kind of perturb them a little bit with these interactions, they have other enhancers or other ways to try to keep expression of the gene at critical times. That's the best answer I have. But I kind of think this is probably a promoter-promoter interaction rather than an enhancer-promoter. But again, we don't have great evidence for that one way or the other. Okay, so this kind of wraps up the first part of the story where I've shown you how we've generated an Epigenome Atlas in the embryonic mouse brain with a variety of data so that we truly understand how these cells look both at an individual cell level and a global chromatin level. We've identified many what I would call high confidence enhancers that quite possibly are critical for regulation of genes that are important for development of cells from these brain regions. It's really important for me to make this data as publicly available as possible. So in addition to all the sequencing stuff being deposited, we've used the UCSC browser so that anybody can go to this website, search for their favorite gene and look at the RNA expression, the accessibility, the histone modifications, and the high-C data that we have. So we think it's a really useful tool for studying brain development. And I just gave some evidence of how finding these brain region-specific interactions and perturbing them can lead to important insights in gene regulatory mechanisms that possibly have a role in human disease. Okay, so now that we've kind of developed this knowledge and these strategies, we're pretty well positioned to understand how perturbations of specific aspects of genes or gene regulatory mechanisms can affect cell fate in disease. So we've basically kind of been going through this type of pipeline to assess this, where we have a mutation or perturbation. We can look at chromatin organization and transcriptome at the single cell level. We can assay cell fate. We can look at cell function with our collaborators performing electrophysiological experiments, and then ultimately animal behavior. And I'll show you how some of these perturbations affect animal behavior and how it might relate to human disease. And so there's 3 stories depending on time. I'll see how much I can get through. The first one is where we look at perturbation of gene repression using loss of EZH2, and then gene activation, and then a specific disease. So these first 2 are kind of taking a sledgehammer to the whole system. We know what's going on now. What happens if we basically try to disrupt general gene repression or general gene activation, specifically in the MG of these mice? And I'm going to give a little bit of background on EZH2. This is a critical methyltransferase. It's part of this big complex. But what's important to know is that it's critical for methylating H3K27, and this is important for gene repression. And so by blocking or perturbing any aspect of this complex, you're going to disrupt normal gene repression and can lead to aberrant gene activation. Now, why this was also an interesting target is because there have been a number of studies that have looked at the function of EZH2 and brain development, and we see that in the cerebellum, loss of EZH2 changes cell fate. It can change the types of interneurons you get. It can also change migration patterns. And so this, the role of EZH2 has been studied in brain development, but not particularly in forebrain interneurons. And so because these cells undergo complex migratory behavior, critical for cell fate, we thought this was a nice target to disrupt gene repression in the MGE and look at how that kind of sledgehammer approach affects cell fate. So we use an Nkx2.1 mouse. So again, this is targeted to the MGE with a floxed EZH2. And what you can see here in red is the normal EZH2 expression, and then the knockout, that expression is strongly downregulated, specifically in the MG, but is maintained intact in the LG and cortex. And again, the readout, this gene is critical for methylating this H3K27 residue, and we see a pretty strong downregulation in the mutant of this histone modification. So then what we did is we took these mice, harvested the brains in the adult, and then we could perform immunostaining. So SST and PV are markers for 2 interneuron subtypes that arise from the MG. Basically, 95% of cells in the cortex that are born in the MG express either SST or PV. So these are essentially non-overlapping cell groups that encompass all MG cells expressed in the cortex. And so what you see in the knockout here is we see an overall decrease in the number of tomato cells. So tomato is just a marker for all MG cells. This is just a reporter. So maybe you can tell here, but there's about a 20% reduction or so of all MG cells in the cortex. But we actually saw a different effect in cell fate. So if you look at the SST in green here, there's actually an increase in the amount of SST interneurons, and then a corresponding decrease in the parvalbumin cells. So this is a fate switch. We're producing more somatostatin cells and fewer parvalbumin cells due to this mutation, specifically in the MGE. I won't show you the data. There's also interneurons that are in the striatum and the hippocampus, and we saw a similar fate switch where more SST and decreased PV cells. One thing I'll note is that the Nkx2.1 is expressed in these cycling cells. We use a different Cre line to knock out EZH2 specifically in postmitotic cells, so after these cells are born, and we do not see these changes in cell fate. So this tells us the function of EZH2 is critical in these cycling cells, and once the cells become postmitotic, removing EZH2 in those cells does not change cell fate. So with our colleagues, Yun Lee at NIMH, they performed some electrophysiology recordings to see in these mutants is the physiology of these cells altered. And so basically they looked at about 20 or so intrinsic properties of these cells, and there was very, very little differences. But what they did notice striking changes in the morphology. So these are some parvalbumin, some basket cells. You can see the wild type here. And if you focus on the blue color, these are these axons. You can see that there seems to be more axons, more branching, and that's quantified over here. So what this means is that in this mouse line where you have fewer PV cells, the PV cells that remain have greater axonal projections and greater targeting and presumably greater synapses. So we view this as a compensation mechanism. How does the brain try to compensate for fewer inhibitory interneurons? One way to do it is that the ones that survive, they have more processes, contact more cells to try to increase inhibition. And then what we want to do is perform multiome analysis. So in addition to looking at changes in cell fate, we can kind of understand what's going on genetically and gene regulatory mechanisms in these cells. So at this point, multiome was available. So this is single cell, ATAC, and RNA in the same cell. So we perform multiome analysis from wild-type headed knockout mice on 2 different embryonic ages. Again, we can kind of look at gene expression profiles so we know the trajectory. So these radial glial cells are up here, and then post-mitotic cells are kind of near the bottom. And then what we want to do is see, are there signatures in the embryo that are indicative or causative for these changes in cell fate? And so if we look at SST here, and again, there's more SST cells in the adult mouse, we see an increase in SST expression here in the knockout mouse at E15 compared to the wild type. Conversely, parvalbumin is not expressed in the embryo. That marker doesn't come on until postnatally, but there are a couple genes, MEF2C and MAF, that are predictive of parvalbumin cells. And what you can see here is that in the wild type, you have pretty strong expression of both of these cells, and then in the knockout, you see downregulation of these. And in addition, because it's multiome, we can look at the motif, so the binding sites of these transcription factors in accessible regions in our DNA, in our cells, and you can see, again, a decrease in the motif accessibility in the knockouts compared to the wild type. So now we can identify in the embryo some genetic logic as to why we see these cell fate changes with increased somatostatin already present at E15 and decreased markers of PV cells already present at also at E15. We can do kind of unbiased comparison to look at genes that are upregulated at these different ages in these volcano plots. And again, you see 2 genes here predictive of PV cells that are wild type enriched, meaning downregulated in the knockout. And then SST over here is strongly enriched in the knockout. There's a fair amount of other genetic analysis we've done with this, but I don't want to belabor the point too much. And so basically the take-home message is that we see kind of a fate switch in this mouse, and also we see changes in gene expression in the embryo that are almost certainly causative for that fate switch. Okay, now I'm going to jump to another story. So that was looking at gene repression, and this is, oh no, sorry, I forgot I had this slide. So in addition to the multiome data, we also perform cut and tag to look at differences of this K27 methylation. Again, this is the target of ECH2. And just to kind of highlight the major take-home message, we found that in this knockout, the changes in the K27 methylation was not equivalent across the genome. We saw some loci that were more susceptible, such as this FOXP loci here. You see this level of K27 methylation in the wild type, and you see a really strong reduction in multiple replicates in the knockout. So this is a loci that seems to be extremely susceptible to this manipulation. Conversely, Nkx2.1, which again is critical in the MG for interneuron, you can see a relative increase in the level of methylation. So this doesn't mean that it's more methylated than the wild type, it just means it's more resistant. The methylation was not removed because all of this is relative. And so this kind of goes back to your earlier point. I think there's evidence that Nkx2.1 has some sort of, because of its critical role in fate, I think there's some evolutionary conservation that has caused this loci to be more resistant to various changes, because despite knocking out a gene critical for methylation, it seems to try as best it can to refute this perturbation and keep high levels of methylation here so that it can be repressed or activated in the proper way. Okay, so that was gene repression, and now I want to talk a little bit about gene activation, but also why this particular mode of looking at gene activation is critical. So methylation at H3K4 is critical for gene activation, but there's a lot of evidence through years of work that have shown that dysregulation of this loci and this methylation is associated with a variety of neurodevelopmental diseases, schizophrenia, also drug dependency. And this is kind of a nice summary of a host of genes that are epigenetic machinery that can either write or remove epigenetic marks. And I'll just highlight 3 genes here. All 3 of these genes target methylation of this particular residue and all have various symptoms and drive human disease. So we think a better understanding of how this particular loci gets methylated is critical for understanding gene activation, as well as maybe kind of some of the underlying causes of various human diseases. Now, what makes studying this complicated is that unlike the previous example where EZH2 is the primary methyltransferase that targets that histone, in this case you have 6 different enzymes that can methylate K4, that can methylate the K4 residue. And so these are expressed oftentimes in overlapping cells and can have overlapping compensatory functions. So simply knocking out one of these is sometimes not sufficient to have a robust effect. Now, we were lucky that a colleague here at NIDDK, Dr. Nkx2.1, had developed a mouse where he mutated the lysine 4 residue to an M methyl, I forget, K4M. And basically what this does is blocks the lysine, so this lysine can never be methylated because it's not there anymore. And it's Cre-dependent, which allows us to express this mutated histone in the cells we want. And what's nice is that this is a transgenic, so basically you have, in a K4M homozygous mouse, you have 2 wild-type alleles, untouched, and then elsewhere in the genome where this was inserted, you have 2 mutant alleles. So this is basically a functional het, which is nice because it kind of mimics human disease, where you have a knockdown or mutation in an enzyme that affects this, and you're looking kind of at a loss, not a complete loss of function, but a knockdown. So we think this is a cool tool to basically have a homozygous mouse as a functional het to study manipulation of this residue. And so what we see is that in these homozygous mouse, they're actually very underweight early on. A number of them die, about 50% of them die by 20 weeks. Their brains are also smaller, which you can see here. The cortex has decreased thickness. But what's interesting is that the ones that survive after about 12 months become quite obese. They can be double the weight of their counterparts. And again, I won't go through this in much detail, but we're pretty confident this is due to perturbations in the hypothalamus, where Nkx2.1 is also expressed. Hypothalamus is critical for feeding behavior and a variety of other hormonal behaviors. Now, if you look at changes in the interneuron counts, what we see is that there's a decrease in interneurons in these mice, MG-derived interneurons, and a strong decrease in parvalbumin, and a slight decrease in somatostatin. So in the EZH2 mouse, we saw a fate switch where one went up, one went down. In this mouse, we see a decrease of both cell types. And this is even more striking in the hippocampus, where if you look in the wild type, there's lots of tomato cells here. And then you can really see these huge pockets where there's very few interneurons in these mice. And we've quantified all that as well. Now I want to kind of show you a video of these mice. So this is a knockout mouse here. These mice have spontaneous epilepsy. You can see these front paws having this back and forth movement, and then it kind of starts to have these rapid movements. This is an example of a mouse undergoing a spontaneous seizure. Here's another example. This one's a little more severe, kind of a catatonic seizure. It'll freeze up and then kind of fall over. And so these homozygous mouse are very sick in terms of the fact they display spontaneous seizures. And again, this correlates with fewer interneurons. So we can do tests to look at the severity of the seizure. So you can inject a compound that induces seizures and look at seizure threshold. And you see that the homozygous here in red are much more susceptible to this test. They undergo seizures at a much more severe level compared to the blue heterozygotes in the wild type mice. Now, we've also done some electrophysiology on these. I won't go through the details, but if you can look on the left, these are wild type for 3 different interneuron subtypes in the MG, and you can see how the red dots and the green dots and the blue dots are fairly well clustered together. If you look in the mutant, they're much more sparse. So what this indicates is that the actual physiological properties of these cells have a greater variance in the mutant, and in fact, they seem to be much more like have properties of immature interneurons. So not only do you have fewer interneurons in this mice, but their electrophysiological properties are compromised and mimic more an immature state rather than a mature interneuron. We've done a lot of behavior assays on this, and I'll just touch on a couple. This is an open field test. Basically, you put the mice in the kind of this big 4x4 basically plexiglass box, and then with the camera you can kind of monitor their movement and quantify it. This is a wild type mice. They kind of prefer the middle, but they're happy to explore around. In this homozygous mice, you can see that they really avoid the middle. This is a sign of anxiety. They don't want to go in the middle. They want to kind of hug the edge where they feel safer and more secure. So not only do they spend less time in the center, but in particular the females actually move a lot more. They travel a greater distance and also a lot faster. So one way to think about this is that in particular the female mice are just zooming around this box, which is, again, a sign of high anxiety. Another way to look at it, to test anxiety, is to put mice in this light-dark box with kind of a 60 decibel noise, and mice are going to prefer to be in the dark in general, but you see an increase for both male and female of these homozygous mice preference in the dark box compared to the light box. We've also done a ton of other behavior assays to show a wide variety of defects in terms of gait, how they react to sounds, their social interactions and learning and memory deficits. And I will say what's really interesting is that the female mice, as you see here, are often have more severe deficits than the male mice, and I won't go into too much more about that. Now, again, we wanted to perform multiome analysis, and in this case, because the mutation is doing things that we think are critical in both the interneurons and the hypothalamus, we harvested embryonic MGE and embryonic hypothalamus, as well as adult hypothalamus and adult and interneurons in the adult cortex. And this is just kind of how this map looks. We can take out the different subtypes and do all kinds of analysis. And the embryonic ones, we see indicators of all these different hypothalamic nuclei that exist. We can see the two types of interneurons and then the hypothalamus. So I'm just going to highlight kind of two things we've learned from this data that's interesting. One is that if we focus on the P60 cortex, so these are the mature interneurons, we can again look at differentially expressed genes. We can look at the different Go terminology factors that they have. But what's really interesting to us is that if you take these most differentially expressed genes up here and then map them onto various human diseases, what you see is a lot of hits on epilepsy, electroclinical syndrome. And so this is really interesting because a lot of the genes that are most differentially expressed in these mutant mice are associated with epilepsy, which we obviously see that these mice have. And so that's one way we can use this data to kind of correlate changes in gene expression with disease. If we look in the adult hypothalamus and we break it down in between cells that come from the wild type and homozygous mice, so we can identify neurons, oligos, astrocytes, and tenocytes. I think what you can appreciate is quite a distinct difference here in the astrocyte cluster, where the mutant and the wild type mice are clustered pretty differently. And we can, again, extract specifically these subsets and kind of look at the differential gene expression, and that there's a whole population of astrocytes that are basically missing in the homozygous mice in terms of gene expression and function. Another thing that was interesting is that if you look at the proportion of cells, again, astrocytes are blue here. Based on this, it looks like there's a lot more astrocytes in the mutant compared to the wild type. Is that true? In fact, it is. When we do immunostaining for this marker of astrocytes, we see a lot more astrocytes in the mutant compared to the wild type. So this isn't something we would have predicted. It's not something we would have had any idea about, but it looks like this mutation causes a significant increase in astrocytes and possibly a loss of a particular subset of astrocytes in the homozygous mouse compared to the wild type. Now, tenocytes are a really interesting group of cells, even though it's a very small population here. They're critical for the blood-brain barrier. So the hypothalamus is a region where cues from the brain can enter the bloodstream and have crosstalk in general. In the rest of the brain, there's significant restriction for anything to cross the blood-brain barrier. But this hypothalamus region, and in particular these tenocytes, are critical for where the brain can communicate with the bloodstream. And it has a variety, they have a variety of function and roles and variety of functions that we think are affected in these mice. So we took cross-sections to immunostain these regions with GFAP, so the astrocyte marker, and vimentin, which marks the tenocytes, and everything here. There's pretty good organization in terms of the cells. If you look in the mutant mouse, what you can see is striking disruption of this organization. There's way more NKX cells kind of in a disrupted manner here. These astrocytes, for some reason, are on the edge and squished and not in the middle. And you can see really striking differences in these tenocytes morphology. So there's clearly something going on that's perturbing the normal organization of these cells at this critical area important for blood-brain barrier and communication between the blood and hormones in the brain. And so this, again, all fits into why these mice possibly have obesity and perturbation of behavioral deficits and feeding deficits. Okay, so I'll run through this last part very quickly. So the last two things, again, we're kind of taking a sledgehammer approach. So now we want to look at genes that really are more affected, more correlated with human disease. And one is called this gene ARCS, a transcription factor. This has been well studied in the literature. It's on the X chromosome and is responsible for about 10% of X-linked intellectual disability diseases. You can see there's a ton of point mutations in this gene. And more recently, since it's X-linked, it was thought to primarily or only affect males, but there's recent evidence now that actually female carriers, so hets, also can have mild and severe phenotypes. What's interesting for us is this gene is known to be expressed in the ganglionic eminences, and studies have knocked out this gene in post-mitotic cells, but there hasn't been much studied when you knock it out in recycling cells. And so we generate and use our Nkx2.1 creed to knock out this gene in the cycling cells, so we can see how that affects interneuron fate, but also it allows us to look at how a heterozygous mouse affects a female, because that hasn't really been studied. So just to run through this data pretty quick, the males are very sick, they're very underweight, they pretty much all die by 8 weeks, whereas phenotypically the female hets are normal size. So again, males are only knockout because it's X-linked, females can only be het, again, because it's X-linked. And here's some videos. So these, the males are very sick. Here's infantile seizures, these spasms you see even when the pups are about 8 weeks old. Again, this should look familiar from the K4M mouse, and these mice are also hypoactive. We put them through the same seizure score. Again, only the females, because the males die, and even injecting the drug causes them to die. But we see the females have a higher susceptibility of seizures. And again, we're currently putting them through a battery of assays. What you can see is really striking loss of interneurons. So this is the cortex hippocampus. This is a normal amount of MG-derived interneurons. The female hets, again, have a decrease. And then the male knockouts really have a striking loss of interneurons. That can be seen here, and again, quantified, where you have just a striking loss, 80 to 90% loss of interneurons in this mouse. And this is more severe than the other studies that knocked out ARCS post-mitotically. They didn't have nearly this amount of cell loss. One cool thing that we're trying to integrate into the lab is spatial transcriptomics. So all the single-cell data we've done is great, but with spatial transcriptomics, you can look at spatial information in terms of gene transcription and differential gene expression, as well as look at non-cell autonomous effects. So this is just kind of some initial data we've had for a couple weeks, looking at the wild type in the heterozygous mouse, where what you're doing is, rather than having a single cell resolution, you're looking at a spatial region. So you can look at gene expression both in terms of the spatial map as well as in a UMAP plot. You can look at gene expression profiles. So again, here's the wild type, and in this case, the het female. We're still doing this in the male, but you can see a strong downregulation of parvalbumin and GAD1 and LHX6. So we're trying to integrate both the single cell data and the transcriptional data to really have a comprehensive way to look at both spatial information and cell autonomous effects with our single cell and cell non-autonomous effects like this. Okay, so just to summarize, I've showed you how we've developed an Epigenome Atlas with a multimodal approach to really characterize these cells in the embryonic brain as they're making these critical brain decisions in cell fate. I've shown you two approaches we've used to kind of take a sledgehammer to gene repression and gene activation and show how that affects both the fate, either percentages of cells and cell loss, and in some cases really striking deficits in animal behavior. And now we're well positioned to kind of look more specifically at genes we know are critical both in interneuron development as well as human disease, and kind of take this pipeline to really hammer home and hopefully provide important insights into some of these diseases. So I hope I've kind of convinced you how we're trying to use a variety of approaches to really understand how you go from these kind of heterogeneous population in the embryo to this incredible diversity in the adult brain, how genetic factors and epigenetic and gene regulatory factors combine with the brain environment to influence fate and maturation of these cells, both in normal development and most critically what goes wrong in various diseases. So just want to thank everybody in my lab, in particular 3 postdocs highlighted up here that were involved in the various projects I talked about, a variety of postbacks, important collaborators, particularly Pedro Roca for his help with epigenetics, the McBain lab for and Sion Leeds lab for electrophysiology, and Ryan Dale runs the bioinformatics core that we really couldn't do a lot of this bioinformatics without. Okay, that's it. Happy to take questions.
Tim, can I ask you a question about your work on disruption of H3K4 trimethylation? Did you do it specifically for the Nkx2.1 locus,
Yep.
or it was done across the spectrum of all genes?
So this is just so it's a floxed allele, so it's just Nkx2.1. So we just use Nkx2.1 Cre, which is, again, expressed in the MGE, hypothalamus, lung, and thyroid in development. So the mutated allele was only expressed in those regions. So we did confirm that. The mutant allele is HA tagged, so we confirmed both a reduction in the K4 methylation in those, not in all those, in the MGE and hypothalamus, and also confirmed an upregulation of HA specifically in those regions. So my prediction is if we did this globally, and actually the people that made the mouse did it, they used a different Cre driver, but it affected more cells, and their mice died very early. So I think, again, the wild-type allele is intact, but I think upregulation of this mutant allele across the board, like with a pan-Cre driver, would almost certainly result in animal death, which the group that developed this mouse line saw using a different Cre driver. So.
And I assume that by doing that, you effectively downregulate the activity of all the enhancers of Nkx2.1. And if you would like to go into the granularity of the regulatory program of that gene, you can probably somehow go one by one and CRISPR them out or something like this and see how you were talking about this evolutionary ways of stabilized gene expression. So that probably involves these shadow enhancers, synergetic activity of multiple of them. So in order to decipher that inside of Nkx2.1 locus, I guess something like CRISPR would be needed to break that.
Yeah, so
into
what
pieces.
the last piece of data we're waiting on that story is actually cut and tag with the K4M trimethylation so that we can look at differences throughout the genome. Because again, if this Nkx2.1 is more resistant, as you were hinting at, maybe the Nkx2.1 locus is going to show greater resistance to this perturbation than other loci. And so that's kind of the last piece of data that can speak to that we're kind of waiting on right now. But there are other ways that you can kind of manipulate that with CRISPR or other ways to look at that effect. I mean, it's a little bit tricky too because again, 50% of these homozygous mice die, so we're looking at the healthiest of the sick mice that survive. And because theoretically it's a 50-50 ratio of whether the mutant allele is there or the wild type allele is there, it might not be 50-50. It's very complicated to look at that, but it's a dirty mouse in the sense that there's going to be variability just theoretically in which allele is integrated into which site. And some sites might have the mutant allele, and those sites might be more critical for particular gene activation and thus give a higher phenotype. So the mouse model is really nice for some things, but it is sort of dirty in a sense of having that level of granularity.
Many more questions online, and I would like one more.
Yes, I was wondering, I may have missed some of your observations, but you said you do a knockout in the experiment in which you do the knockout of the enhancer of the STCH2. It seems like at the end you get a different proportion of the three types of cells. Mm-hmm. And I was wondering, where is this effect coming from? Is it coming from a change in fate of your cells? Is this coming from a different cell migration and differentiation? Is it coming from cell division that change the proportion of these cells? Or is this coming from cell death that affected, these cells differentially so that you end up with this differential?
That's an excellent question, and you've hit on almost all the possibilities, I think. So we haven't nailed that down. What I will say is we looked at cell death, and we didn't see significant changes. One nuance in development of these PV and SST cells that I mentioned is that these cells start to be born around E11-ish, and they're basically continue to be born until about the animal is born at about E18. So you have a range of about 7 days that's producing these interneurons. Now, the PV and SST cells are not produced equally over that time. So in the beginning, there's kind of a burst of somatostatin cells, and then their production goes down over time during embryogenesis. Parvalbumin cells are kind of a little more constant. So if you look at E12, E13, you're kind of making a 50-50 mix of SST and PV cells being born. If you look at like E16, it's more like an 80-20 mix of parvalbumin cells over somatostatin cells. So what this means is that in development, if you have a mutation, like I think is happening in this ECH2, where you're kicking cells out of the cell cycle too early, what's going to happen on that developmental timeline is if you're making too many neurons too early, there's a bias for those to be somatostatin cells. And if you're depleting the progenitor pool later on, so you have fewer cells being born later on where PV cells dominate, you're going to have fewer PV cells. So we've looked at that a little bit. The data wasn't as convincing as I hoped, but my best guess, because a little bit of what we know about what ECH2 is doing in cancer, is that these cells, too many cells, are being born too early, which is depleting the progenitor pool. And because of the time course of how somatostatin and PV cells arise, that explanation in and of itself is sufficient to, one, have a decrease overall in the number of interneurons that we see, but also kind of have this fate switch. So maybe, fate switch is a dangerous word. I think what's more accurate is that increased production of interneurons early at a time when you're biased for somatostatin cells and decrease production later at a time when you're biased for PV cells. And that's how you can get this fate switch appearance, but I don't think the individual cells are switching fate, although we don't have direct evidence against that. I think this other scenario is much more likely for the explanation. Okay. Thank you.